Let us start. First of all, good afternoon, everyone. Thank you for being here with us. My name is Mircea Opriș. I am part of HVD team, and we are supporting immunocap technology in Eastern Europe. It's a pleasure to welcome you all to our fourth webinar in our allergy focus series. Welcome to all of you who have joined us in the previous sessions, but also to our new participants. And let us simply mention our previous topics because there is a lot of interesting and valuable materials that you can find in them. And maybe there is interest of any of you that would like to review some of these. So our first session was dedicated to food allergies. We continued in our second session to spring tea allergenic molecules. Our third session was we approach molecules of grasses. And now in our fourth session that will take place today, we have dedicated this to the molecules of weeds and venoms. So for those that are attending for the first time our series of lectures, we are delighted to have you with us. I'm proud to say that this series is conducted by Professor Rudolf Valenta, and all of these are designed to provide valuable insights in the key allergy topics that we hope it can be a value in your everyday clinical practice. But moreover than this, our main objective is, of course, to improve our patients' quality of life. So if you'd like to review or follow any of our future webinars, once you're registered, you will be automatically invited to our next sessions. And it's important that you don't need to do any further steps. As I said, I see a lot of participants comparing to what we have been expecting. We know it's summertime, it's difficult for all of us to reach each other. So also in the context of holidays, I'd like to take this as a very good feedback that your presence here is in a big number, and we thank you for that. I really think that it means a lot for us. We understand that we are providing valuable information to you, and we are happy to do it, and we will keep doing this in the future. Once again, it's an honor to introduce Professor Rudolf Valenta, world-leading expert in molecular allergology from the Medical University of Vienna. His pioneering research has significantly shaped our understanding of the allergic disease, and I find this a privilege to have him here today with us to learn from him. As always, I encourage you to submit questions. Professor Valenta will try to answer to each of them by the end of the webinar. And just a short reminder, this webinar is recorded for the educational purposes. It would be available for further watching for our participants which they would like to have any review of the material and also for previous sessions. Now, with further delay, let's welcome Professor Rudolf Valenta, and thank you again for joining us today.
Thank you, Mircea, for the nice introduction. Yeah, and I have to say, for me, it's always a pleasure for me to share a little bit of my knowledge and also some very late findings with you. In the end, the goal is always to make diagnosis and treatment for allergic patients being better. And this time, I'm some sort of dipping into the summer. We can almost say a little bit a late summer. The summer is the time of the ending grass pollen season, and it's the beginning of the wheat pollinosis, depending on where we are sitting and what the climate is. And at the same time, we're encountering something else that sometimes can be unpleasant, and this is bees and wasps. So we have to deal with venom allergy. Now, my first slide is kind of thought to provide you my email address. The reason is that you are always welcome to write me direct emails if you have questions and any things you want to know. I have listed for you here also some useful homepages about our research programs. And you can find information, of course, about the publications in Scopus and in PubMed from us. And please note, we have a very strong allergy research program at the moment in Austria. It's called the Danube Allergy Research Cluster, where several research groups focus on very severe and life-threatening forms of allergy. So I think the presentation is very timely. Now, let's jump immediately into the world of the wheat-borne allergens, and we can also say in the world of all the allergen molecules. And again, I would like to emphasize that there is a very important resource for you that you all can access by your computer from home via the internet. And this is the allergen nomenclature database. The allergen nomenclature database was founded long time ago in the late 70s, and it's a collection of all the allergen molecules and the description that have been found so far. And they are categorized in this homepage that you can access by this link, so that you can search by allergen name, but also by allergen source. So for example, you can put in here ragweed, or you can put in here mugwort, and then this will bring you to a next page where you can see the list of the ragweed or the mugwort allergens. And there is also links then to the sequence and to the publications that are concerned to this. And I think this is a very important resource because it connects the previous world of allergen sources with the current world of allergen molecules and the knowledge of it. But of course, you're also welcome to read some special reviews. And I'm also mentioning here for you some of the important review articles that can help you further in the knowledge about the allergen molecules. And for wheat pollen, I would like to emphasize this very nice review by Gabriele Gardemeyer. It's some time ago that it was written 10 years ago or more, but still it's a very complete and very timely summary of the field. Now, as we go into wheat allergen sources, I have made here a list of those wheat allergen sources that are particularly important for us here living in our geographic area, but also beyond. And I would like to discuss them point by point with you now in the lecture in a systematic form. There is ambrosia, this is ragweed. It's a very potent allergen source, highly allergenic. There is mugwort, Artemisia vulgaris. Of course, there is also other species. There's Chenopodium album. There's Salsola kali, saltwort. There's Plantago lanceolata, maybe of less importance. And there's Paritaria, a very important wheat allergen source in the Mediterranean area, for example, in southern Italy, but also in southern Spain, southern France. But we can find this wheat also in the Middle Europe, so it's actually quite distributed. Now let's start with ragweed pollen. Ragweed pollen actually becomes of more and more interest because it's a weed, a plant that is likes to expand. It's like a migrating plant, if we want, that drills more and more into the heart of Europe and is actually causing a lot of problems. In this review written by Kuanchai Wai Chen, you can find a very nice overview about the burden of ragweed allergy in Europe, but also you can find a very comprehensive summary of the ragweed pollen allergens. Now, ragweed is very interesting because it's distributed in the whole world. It's particularly important in Northern America, and it's also spreading in Europe because it's more or less brought by former times ships and also other means of transport to Europe, where the plant is going to expand. And it's particularly important for the eastern parts of Europe, Hungary and Romania, from where it's kind of spreading into a central part of Europe. So it's very important, but of course it can be also found in South America, it can be found in Asia and in other continents. So it's really a very aggressive allergen source. Now, the interesting thing historically is that ragweed was extremely well researched in Baltimore area in Northern America in the United States, where actually in Baltimore there was a very active group of researchers in allergy centered around the clinician Philip Norman, but also Larry Lichtenstein was working there. He was the man who invented the basophil degranulation assay. But there were also other eminent researchers, like, for example, the couple Ishizaka was working there. They discovered IgE. There was David Marsh, a very famous allergologist who investigated the genetic roots of asthma. And at the same time, he was actually also very busy in creating attenuated allergen immunotherapy vaccines. He was actually the inventor of the allergoids that you may know and that are still in use in many, many different areas of the world. And the principle is used by many different companies who produce allergen extracts for actually vaccination. Now, why is Baltimore so interesting? Baltimore is so interesting because it's an endemic place where ragweed pollinosis is very strong. And this is also the reason why the Baltimore people were pioneering many, many immunotherapy studies, but they were also using the ragweed model to investigate fundamental findings in allergy and immunology using ragweed and ragweed allergens as molecular models, if you want. And actually there they also purified the major allergen of ragweed, the AMP1 molecule, and they were also the first to show that immunotherapy with the isolated AMP1 molecule can be very effective. So this is what you should bear in mind. Baltimore is kind of the cradle of ragweed pollinosis. Now, the hard stuff in what we are doing in molecular allergology is, of course, to get an overview about the ragweed pollen allergens. And I mentioned for you the iOS allergen nomenclature database. And you can here find also the table of the ragweed allergens that we will be discussing today in some more detail that are summarized in the review by Gabriele Gardemeyer. Now, without any doubt, the major allergen AMP1, a peptide lyase with a molecular weight of about 38 to 40 kilodalton, is the major allergen in ragweed, the most important. What is also very important to comment on, it exists in different isoforms. Isoforms are variations in the sequence that are not completely identical. And in the case of ragweed, this seems to matter because not all of these isoforms are equally allergenic. The allergen AMP2 was actually then grouped into the AMP1 allergen family as an isoform. And there's also a couple of other allergens that make the system a little bit complicated, if you want. And I will explain you immediately, because there's some very interesting molecules like the AMP4 protein. The AMP4 protein is a defensine-like protein, and it's actually very similar to the major allergen of mugwort. Mugwort is also a very important wheat allergen source, but it's distinct from ragweed. And it, of course, also requires different treatment and different immunotherapy approaches. However, AMP4 and RP1 are kind of very related proteins, so they share a high degree of sequence homology, so they are quite relevant. And it plays an important role actually in the differential diagnosis. The AMP1 is a rather tiny molecule. It's recognized by fewer patients, so it doesn't seem to be so important. But we should pay attention to the AMP6 molecule, and this is a lipid transfer protein, and you will know lipid transfer proteins also as food allergens. This is also mentioned here. But what is very peculiar about the lipid transfer proteins, unlike other cross-reactive allergens like the profilins and the calcium-binding allergens, the lipid transfer proteins have a rather restricted cross-reactivity. So you cannot deduce from the fact that somebody reacts with a lipid transfer protein from one allergen source that this person will have automatically cross-reactivity with lipid transfer proteins from other allergen sources. And I will illustrate this to you by a sequence comparison that will make it easily explainable. Now, like in many other pollen allergen sources, we find profilins and calcium-binding proteins in ragweed, so they are highly cross-reactive with other allergen sources. And there is a newly described so-called major allergen of ragweed. It's called the AMP11 molecule that was rather recently described. And we will speak about this protein in some more detail because its importance as a major allergen is to some extent a little bit not completely explained. And it's not sure if it has really this importance that is given here with the 50 to 66% of sensitization rate. And we will discuss in a moment about it. There is an enolase also in the ragweed. Enolases can be found, for example, also in fungi and in other pollens. And they are quite frequently recognized, but their clinical relevance so far is not well investigated. Now we have, in fact, molecular structures for many of the ragweed allergens. You can see here the AMB1 molecule. It's a pectin esterase. It's quite a complex and not easy molecule also to make as a recombinant protein. So this is an important comment. We can see here the profilin from ragweed and the calcium binding proteins. These two differ from each other only in the number of the calcium binding pockets. The AMB9 is the more common protein actually. It cross-reacts with other calcium binding proteins in other plant pollens. And of course, as I have mentioned here, the other allergen molecules, and we will discuss and jump into it now in some more detail, and I will try to make it as systematic for you as it can be. Now let's start first of all, and this may be written now in our minds with AMB1, the key allergen molecule in ragweed that exists in different isoforms. This means that these AMB1 forms, AMB1 01, 02, 03, they differ a little bit in the amino acid sequence. And this means that they are not completely cross-reactive. You can see this here in this very nice paper by the group of Fatima Ferreira. Actually, only the AMB1 molecule inhibits very completely Ig binding to AMB101, but AMB102 inhibits less, and the isoform 03 even less. So what we learn from this is that there's three isoforms. They are very similar, but nevertheless, they are not completely cross-reactive. And we have to pay attention to this because we need to distill out what will be the most important form for the diagnosis. At the moment, most of the people work with the AMB101 isoform, and this performs very well in the diagnostic approach for the serology. Now we come to a very important point, and I want to bring it just at this moment when we are discussing the AMB1. We have understood now AMB1 is the major allergen of ragweed, and we were always taught and we learned that AMB1 is the leading allergen molecule that indicates genuine sensitization to ragweed. This is true, but we have also to acknowledge that there is a similar molecule in mugwort, the ATV6 molecule, and it's nicely described in this paper also by the Ferreira group in the year 2012. And this protein is actually similar to AMB1, and there is cross-reactivity between these two proteins. And I mention this because it's a little bit of a challenge to distinguish genuine sensitization to ragweed pollen and to mugwort pollen, because in principle it's not as clear like in birch or in grasses, where we have genuine marker molecules. In mugwort and in ragweed, we have actually similarities between the major allergens. So the differential diagnosis is not completely as easy as for grass pollen and for birch. And I will help you with an algorithm that we developed and that will help you a little bit in the diagnostic procedure. You can see here the sequence homology between actually AMB1 and ATV6. And you see here in the upper line, the amino acid sequence of AMB1-3 isoform. And you see below the ATV6 sequence. The stars that are printed below indicate the identical amino acids. And as you can see, there's a lot of stars. And this means that these two proteins are highly related. And this explains also the cross-reactivity between the both of them. So it's very important to consider also the ATV6 molecule for differential diagnosis. Now let's jump to a protein that, in my opinion, is very specific for ragweed, and this is AMB6, the non-specific lipid transfer protein. And this is an allergen that actually should not be neglected. AMB1 is the most important, but AMB6 is also recognized by a considerable portion of ragweed allergic, pollen allergic patients. It's about 30%, so this is not nothing. And what is also very important, the AMB6 is very, very specific for ragweed, and I will explain you in a moment why this is. You can see here the sequence alignment of the AMP6 molecule is on the top line always. And below we can find all the lipid transfer proteins from many different other plants. Now the fact that we print here all the amino acids indicates that all these other lipid transfer proteins are not as similar as one would have thought. This means that there's a very limited sequence homology identity with the AMP6 molecule. And this is also the reason why AMP6 can be somehow considered as a quite specific marker for actually being sensitized to ragweed pollen. Of course, not every patient reacts with it, but if you have a patient that is positive with AMP6, it's a very good evidence that the person is generally sensitized to ragweed. So consider this protein, please, also in addition to the AMP1 molecule. This is a genealogical tree that illustrates the sequence relationship. And what is quite interesting is that the AMP6 molecule on the top is quite far away from the ATV3, that's the lipid transfer protein from mugwort. And this means that these proteins are not very similar. And you will also see that very important lipid transfer proteins from example from fruits, like the ProD3, are very distinct from the AMP6, and also the parietaria molecules are quite distinct in their sequence. So lipid transfer proteins are not the perfect cross-reactive allergens, and that's a very important lesson to be learned. Now, another little point here, and I recommend you to read this review and paper that is written by a Romanian group, headed by Professor Carmen Bănătescu from Timișoara. These people by heart became very active in researching ragweed, and they are driven by very strong motivation. And the motivation is that in Romania, ragweed pollen is very important, and so they decided to work very intensively in research and study the molecular components actually of ragweed. In this paper, they have been looking in the different allergen molecules in ragweed, and their conclusion is that AMP1 is very important, but there's also other important molecules, and another one of those is actually the AMP8, and this is the prophyllin, a highly cross-reactive allergen from ragweed pollen that needs to be considered. It's sometimes positive in up to 30% and more of the patients. So also this protein, this allergen, should not be neglected. Now let me bring you to something that I think is very interesting, because we were all thinking all the time that the prophyllins are so strongly cross-reactive that out of a diagnostic test we cannot deduce much, because they are almost identical, regardless whether they come from ragweed AMP8, from mugwort ATV4, from birch BETV2, or from Timothy grass FLUP12. So traditionally, we were always educated to think that if you are positive with one, you will be positive also with the other. But I would like to highlight now the importance and the usefulness of quantitative Ig measurements, because sometimes you will find people that are only sensitized to prophyllin, and then you will see that actually everybody here listed is more or less cross-reactive to all these different prophyllins. But when we go, for example, a little bit in the details, and we look, for example, here at the patient number 72, we will easily recognize that this person has the highest specific Ig levels for the ATV4 molecule. And this means that obviously the mugwort was a genuinely sensitizing allergen source for this patient, because the specific Ig levels for AMP8, for BETV2, and for FLUP12 are lower. So this means that when we apply quantitative measurement of Ig levels to prophyllins, we can also get an indication what the sensitizing allergen molecule might be. And here Ig levels are very, very important. Another example, you can look at patient number 41. And this patient is obviously sensitized by birch, because here the BETV2 is the one having the highest specific Ig levels. So what we learn from this is that there is cross-reactivity among the profilins, but they are still useful for differential diagnosis, in particular when we consider the allergen-specific Ig levels. They can give us another hint what is the sensitizing allergen source. Now we come to something that somehow has shaken up the scientific world, especially in the field of ragweed pollen allergy, and that's actually the reporting of AMB11, a cysteine protease, as a potentially important ragweed pollen allergen. And this work comes from the Staroshen research group headed at that time by Philippe Moinchon from Staroshen. And they were actually purifying the AMB1 protein from ragweed pollen by a procedure called two-dimensional electrophoresis and cutting out of dots that were Ig reactive. And what they were doing is they were sequencing these dots and found actually this cysteine protease. Now, what you must know is, and this is important to understand this paper, that the AMB11 is in terms of the size, the molecular weight, not so much different from the AMB1. So it's very difficult actually to isolate the dot that is only AMB11, and there is no contamination with AMB1. And what I want to say is that with this protein chemical approach, it's very difficult to differentiate between a protein that is AMB11 and this AMB1. And that's also the problem. What we have is when we speak about the relevance of AMB11, because up today, we do not really have a good recombinant AMB11 that can be used to prove that AMB11 is really an important ragweed allergen. Having said this, there is more, of course, investigations ongoing. This paper, again, by the group of Professor Buno, has investigated AMB11. And when you look in the detail, they find rather low Ig levels against AMB11. And again, it's recognized by many patients, but the Ig levels are very low. So we have to be sure about whether it's really clinically relevant. So I think at the moment we can disregard it to some extent for the differential diagnosis. Now I have brought here for you a little bit of an exercise sheet. It's a sheet from one of our recent studies where we test asthma patients who are sensitized actually to mugwort and ragweed pollen. And it's a very nice thing because you can see here the different allergens. You have here the ATV1, the major allergen of mugwort, and the homologous protein from ragweed, the AMB4. So we just can compare the both of them and also the Ig levels. We have here the ATV4 and the AMB8. These are the two profilins from mugwort and from ragweed. We have here the calcium binding protein from actually mugwort, ATV5 and AMB9 from ragweed. We have here the major allergen of ragweed, AMB1, and the corresponding protein from mugwort, ATV6. And we have here the lipid transfer protein from mugwort. Now, when we do the exercise and we look at patient 290, it becomes very clear that this patient is genuinely sensitized to mugwort because this patient reacts only with the lipid transfer protein from mugwort, but not with any other protein, so not with the major ragweed allergen, as you can see here. Interestingly, when we turn now to the other couple between mugwort and ragweed, we can see here a patient that is exclusively sensitized to the ATV1. So this is bona fide, a mugwort sensitized patient. But when we go down, we can find also another patient, that's patient 691, that has higher Ig levels to AMB4, and at the same time is sensitized to AMB1, as you can see here, but not to ATV6. And the Ig levels actually to the calcium binding protein in the profilin from what we can see here, ragweed are higher. So we can conclude that this is a genuinely sensitized patient to ragweed. So this is a very nice example of how we can handle the differential diagnosis. And it's also very nicely reflected here for example, by this patient, higher Ig levels to ATV1 than to AMP4, actually higher levels to ATV4 than to AMP8, the profilins. So here, again, a clear diagnosis, this is a genuine mugwort sensitization. So what I want to say is when you are trained in this and you can a little bit work with Ig levels on the one hand and the specificities, it's a real fun to do differential diagnosis of mugwort and ragweed pollen allergy, but it is not quite as easy as for grass pollen and for birch and for ash. We can continue this little exercise here, as you can see. So we can find here different sensitization scenarios. And I hope I could make you a little bit fun out of this here in molecular allergology. Now, why are we doing that all? We are doing this because we have a very effective form of treatment, and this is allergen-specific immunotherapy. And again here, the group of Professor Puno is spearheading the field. I like this very much because Romania is now really dominating the molecular research in the field of ragweed pollen allergy because they have this genuine effort to do it because they have the patients. And they also have the issue, what specific immunotherapy should they prescribe to their patient? So in order to do this, they performed a study, a very nice study, where they were looking at commercially immunotherapy vaccines that are available at the moment in Europe that you can buy from different companies. And we have here different preparations that I have listed here that's close to it. That's Tyrosit, that's Polinex, and that's a vaccine from Diatea, a company in Spain. And actually, the group of Professor Puno has investigated the capacity of the different vaccines to induce IgG antibodies against AMP1 by immunizing rabbits with these immunotherapy vaccines. And as we know, the rabbits behave quite similar as the human. So we can see here nicely what you can expect to happen in the human. And what you can see here is that actually the Diatea vaccine is giving you the strongest IgG response against all the three AMP1 isoforms. Whereas, for example, Polinex is very, very poor in inducing IgG antibodies. The same for Tyrosit. So these are not very good vaccines, actually, in our opinion, for ragweed pollen allergy. Close to it, to some extent, works, but it's less effective in inducing the IgG antibodies as compared to the Diatea vaccine. So what we learn from this is that different ragweed immunotherapy vaccines have a different capacity to induce IgG antibodies against the major allergen of ragweed, the AMP1 molecule. Now, the group of Professor Puno has also investigated the capacity of these vaccines to induce antibodies against the other ragweed pollen allergens. And what we can see here that also here we have a much lower but still distinct response induced by the Diatea vaccine against AMP4, AMP6, AMP8, and AMP11. However, several of the other vaccines fail almost completely in inducing IgG antibodies against the other ragweed allergens. So you can see here, it is very important to actually do the molecular testing because not every vaccine is suited as well. And you can also see that some of these vaccines are not performing very well for actually the treatment of ragweed pollen allergy. So for me, it's very clear I would prefer the one that induces the highest IgG antibody levels because they are also supposed to be the blocking antibodies. Now, in this paper, we have also thought about an algorithm for ragweed pollen allergy. And so far, we have been mainly looking for differential diagnosis of ragweed and mugwort for AMP1 and for ATV1. But we have learned now that the situation can be a little bit more complex because AMP1 is similar to ATV6 and ATV1 is similar to AMP4. So we have to consider all the four molecules in the algorithm. And it's very clear when you have a positive test result and the Ig is higher to AMP1 than to ATV6, this is reflected here, you have a case for genuine ragweed pollen sensitization. When you have positivity for AMP4 and the Ig levels are higher, or there's only a sensitization to AMP4, then again you can confirm liquid pollen sensitization. Cross-reactive allergens like the profilins and the calcium-binding proteins may help you in this decision. Again, when AMP8 levels are higher than the other profilins and AMP9 levels are higher, you can again confirm liquid pollen sensitization. So this algorithm is very nice. I highly recommend you to have it and to use it in the practice. It's at the moment the best that we can have. But of course, later on, there will also appear, of course, the lipid transfer proteins in this, and they will also play a role in this algorithm. So actually, molecular diagnosis is getting more and more precise by the time. Now let's move to Artemisia. Artemisia is very, very frequent in all of our countries. I think there is no country where it does not exist, and it's very important. And in principle, we have in Artemisia some key allergen molecules that are important. It's the major allergen ATV1, and it's also of importance, the ATV3. And what is very interesting, the ATV3, the non-lipid transfer protein, is actually a protein that may vary in sensitization capacity in different countries. And this may have to do with the occurrence of different mugwort species. Of course, we do have also profilin and calcium binding proteins in it. So we have to consider it, and we have learned now that there's a ATV6 that is similar to the AMP1, so also this needs to have some consideration. ATV1 is very well investigated. Its structure is known. Again, Fatima Ferreira was pioneering this research, revealing also the structure of the protein. It's consisting of basically two domains. So it's the defensin domain. It's like a head. So it's like a little head that you have here. And then you have a tail, and that's the proline-rich domain that is shown here. So this is the head domain, and this is actually the tail domain. What we know so far is that major IgE epitopes sit in the head domain of this protein, and this is also a very stable protein because it contains several disulfide bonds. So you can boil it even, and it still will stay in some shape and remain allergenic. Now, actually, mugwort is very interesting because it's not only a big health concern in Europe. Mugwort is also very important actually in China. In China, we have huge areas in the northern parts of China where Artemisia is dominating. Artemisia is also dominating in Kazakhstan and is also very common in Uzbekistan and in Mongolia. So it's really a very important wheat allergen source. But as I said, you will not find any country in Europe where there is not Artemisia pollen allergy. Accordingly, researchers from China, among Professor Gao, have been very active in this field. And one take-home message that I wanted to send to you here at the beginning is ATV1 is a very good marker for mugwort pollen sensitization, but please consider also the AMP4 because they are similar. So there is one kind of a brother or a sister of ATV1 in the ragweed. So when you make the final diagnosis, please consider also the sensitization to ragweed. Now, Professor Gao has published several papers, and I find them very illuminating and very interesting because you can see here that ATV1, of course, is the dominating molecule recognized by most of the patient. It binds the highest Ig levels. The ATV2 allergen doesn't seem to be very important, so we can almost neglect it. It's low Ig levels, but ATV3 can be very important. And what is also very interesting, there is another molecule on the horizon that's at the moment discovered only in one Artemisia species, but not yet in Artemisia vulgaris, but surely there is a cross-reactive allergen. So we must consider in the future that there will be an ATV7 molecule, and according to our test results, it can be a very important allergen molecule that will need more attention actually in the future. Now we have understood that mugwort and ragweed pollen allergy is a little bit more complicated than AMP1 and ATV1. the world is more simple for the other wheat allergen sources like Chenopodium album, because here we have basically one major allergen that is very specific for Chenopodium, and this is KR1. It's a OLE1-like protein. OLE1, you know, is a protein in olive pollen. But the good thing is that KR1 has a very low sequence identity with OLE1. So when you find it positive, it usually indicates a genuine sensitization to Chenopodium album. Sal solar kali is actually a plant. You can see it here. It's a tistle that grows basically in very, very dry areas, basically in areas that almost like a little bit like a desert. It's the salt tistle or saltwort. And here everybody so far was looking at salt K1. It's a pectin methylesterase family protein, a little bit similar to AMB1 if you want, but quite specific for actually saltwort. But recent findings also from us indicate that there's also another protein, and I'd like to share it with you in a moment. In the salt tistle, we have also an OLE1-like protein, it's salt K5. So far, we have been relying very much on salt K1 for the differential diagnosis of sal solar kali sensitization. Salt K1 is available as a recombinant protein now. That's also very good because you can have it as a non-glycosylated protein, avoiding CCD positivities. You know, and you remember, we always try to keep our assays free from CCDs because CCDs hide sensitization and kind of create a fog of cross-reactivity. So in all our tests, we want to have mainly non-glycosylated allergens, and we would like to have no glycosylated proteins in it. So this is a good news. We have actually studied also sal solar kali recently, and the results are reported in this paper published very recently in Frontiers in Immunology. And there something quite interesting pops up, and this is that the profilin is actually sal K4 is also a very important saltwort allergen. So I think you should consider it together with the sal K1 in the differential diagnosis of saltwort allergy. Plantago lanceolata is a small plant. I think the pollen burden from this plant is rather low, but still we can find sensitized people, and the leading allergen in this, and that's also available in component testing in the immunoCUP as far as I know, is the PLA1. It's again a OLE1-like protein, but this is very specific for Plantago lanceolata. So this will help you very much to identify Plantago lanceolata sensitized patients. Finally, we come to a very important allergen source in the Mediterranean area, but also in Austria. When I go jogging, for example, in the Vienna Prater, I can see many, many places in the forests where there is paritaria. This may have also to do something with climate change. It gets more and more warm and hot here. So plants from Mediterranean areas come up to our region. And here we have basically two key allergen molecules. That's the PAGI1 and the PAGI2. Both are non-specific lipid transfer proteins. And again, I go against the prejudice that lipid transfer proteins are highly cross-reactive. They are not. PAGI1 and PAGI2 are really genuine sensitizing molecules in paritaria. We have also cross-reactive allergens, profilin, PAGI2, and calcium-binding proteins. But PAGI1 and PAGI2 are the real markers for paritaria sensitization. And there's also immunotherapy available, so this you should be on your menu list to test wheat pollen allergic patients. And as I said, this paritaria pollinosis, as reviewed here in the early 1990s, was considered an issue that is somewhat around the Mediterranean Sea. But in the meantime, paritaria has moved up to Europe, and it's something that should be on our menu list when we look in Europe, and of course also in Eastern Europe and in Middle Europe. Now we have expressed PAGI1 and PAGI2 resembling the allergenic epitopes of the natural proteins in actually eukaryotic expression system. These two proteins are extremely reliable when you combine the two PAGI-1 and PAGI-2 allergens, you get almost as close as with the extract. So this is really very potent and very useful molecules. And there's one important take-home message here. PAGI-2 alone is not sufficient. You should also take into consideration because PAGI-1 is a little bit different in the sequence from the PAGI-2. So it's better to do the diagnosis with both of these molecules. And you can see that when you add them both, you reach the same Ig binding capacity as with the Paria Utaita extract. So now we are coming into the finish, and that's a hot topic. It fits to the summer. And the hot topic is here venoms. Venoms is a hot topic because when we do just serology, we will find many people who are sensitized. And there's also, of course, very effective forms of immunotherapy. But the question to the patient and to the doctor is always, do we have a bee sensitization or do we have a wasp or hornet sensitization? Hornet and wasp fall in the same category. Honeybee and bumblebee also in the same. The problem for the patient is the patient gets stung, it hurts, the patient gets rid of the insect and goes to the doctor, but very often cannot remember, was it a wasp or was it a bee? And then the doctor starts the diagnostic procedure. And then we come into the question, was it wasp or was it bee allergy, or is the patient double sensitized? Now here we have, I think, made a major contribution to the field with this paper. It's about the differential diagnosis for bee and wasp allergy. And in this paper here, we have been introducing the testing with non-glycosylated bee and wasp allergens. And I would like to show you in a table very clearly what the value of this is. You can see here under the term SB patients that are actually sensitized to bee. And what is very important here, you can see that many of these patients also have Ig antibodies against bromelain and against flupiphore. And what is the reason actually that the so-called bee allergic patients react with a plant allergen, the flupiphore, and CCD, a carbohydrate marker? The reason for this is that APM1 contains also carbohydrates. So very often, bee sensitized people have also Ig against carbohydrates. When we express the other bee allergen, the APM2, the hyaluronidase, in insect cells, we also get partly glycosylated protein. And when we look down now here, in the patients that have no bee and no wasp allergy, they are positive to the glycosylated insect cell expressed protein. And they are also, on the other hand, only sensitized to flupiphore and to the CCD molecules, but not to the APM1 that is expressed in E. coli. Now, this makes it clear that we need to have an AP1 that is devoid of any carbohydrates to make sure that the patient is really sensitized to bee. Now, for wasp allergy, the situation is much more simple because we can use, for example, VESV5, and VESV5 is not glycosylated, so we have a very clear differential diagnosis in the system. However, in the meantime, since 2010, we have started to learn a little bit more, and there have been described a couple of more venom allergens. And this nice paper brings you another step closer to this. And at the same time, it says something very important, and I have written this here. When you have Ig antipositivity to the allergens, it does by no means mean that the patient has an allergy. And this is a highly important topic because Ig positivity, like for all other things, never confirms that there is really an allergy that is clinically significant to bee and to wasp. The problem here is a legal problem, because when you have a positive Ig test result to a bee and a wasp allergen, you need to explain the patient what it means. It can be that the patient never had a problem, but how can you exclude that the patient will not have a problem with the next sting. So you're in a kind of a difficult situation. And this is why many clinicians prefer to test for P and wasp allergy only when the patient had symptoms to bee and wasp stings. And this is up to you. You can have different tests. You can have component tests in the immunocup, and you can have, in fact, also tests by multi-allergen testing. But when you have a positive test result, the question is the explanation to the patient. What are you going to do? What are you telling about a positive IG test result? Are you going to prescribe an EpiPen or not? Because the problem is then you have seen the test result and you need to react. This paper here very nicely says one thing, and you can find it when you read it. And it says that IG positivity is not a confirmation for being reactive and having symptoms. The second thing is it provides you a very nice algorithm that you can use for diagnosing genuine bee and wasp sensitization. It's a RPM1, major allergen, the RPM3, 4, and 10. RPM10 is an interesting protein that has been recently discovered. And you have for the wasp, the VESV1 and the VESV5. But it's very important that all these proteins are made without carbohydrates to be sure for the accurate diagnosis. We also have cross-reactive allergens. That's the hyaluronidase, and you have to consider them because it can be that the patient is also co-sensitized or cross-sensitized to both of the allergen sources. And we have actually updated this guideline a little bit in a recent article that we have been writing about molecular diagnosis. I highly recommend it. It's a very comprehensive overview published recently in the Journal of Allergy and Clinical Immunology. In this paper, you find a very nice list also of the known and important bee and wasp venom allergens that you can use for diagnostic testing. There is also a little bit of an information about their clinical relevance and how you should handle them. And you can find also a diagnostic algorithm that more or less relies on the one that we have established in this former paper here. So it's in a sense the same. It's again marker allergens for bee sensitization, RPM1 mainly, RPM10, but also RPM3 and RPM4. And for the VESV1 or VESV5, I do not think that you need a POLD1 and a POLD5 because they are highly cross-reactive. There is, however, also cross-reactive allergens. So there is cross-reactivity between the ones that you have seen here. So please consider the hyaluronidases. They are highly cross-reactive, and this will help you to sort out the difficult problem of what immunotherapy to give to a venom-sensitized patient. But I think this is very helpful. But in the end of the day, please consider IG positive test results do not mean that the patient will experience a severe reaction upon a sting. So now, let me summarize after this not so easy lecture. I understand it, but I want to make it now a little bit simpler and want to tell you that in the summer and in the autumn, of course, there's different weeds that are important. The most important ones is Ragweed ambrosia, Mugwort artemisia, Paritaria and Salsola are very important in certain regions, and in some cases, Chenopodium plantago should be considered. There is cross-reactivity among several weed-borne allergens, and I have shown you this for AMB4 and ADV1. I've shown you this also for the profilins and the calcium-binding proteins. So when you make the differential diagnosis, do not only look only at allergen molecules, but consider also the levels of specific IG. The diagnosis of genuine IG sensitization is important for prescription of immunotherapy. It's especially important for Ragweed and for Mugwort, because here we have treatment forms. There's different immunotherapy vaccines, but not all of them equally well induce IgG antibodies against the important allergens. And IgO activity profiling may help you selecting the best possible immunotherapy vaccines. because you have seen that the capacity to induce IgG antibodies against different allergens can vary. And you can, of course, find out against what molecules your patient is preferentially sensitized and then select the suitable treatment option. Bee and wasp allergy. What is very important is please test, if you can, with non-glycosylated allergens to eliminate false positive reactions to CCTs. We have very nice diagnostic algorithms based on molecular tests that allow you the precise description and prescription of immunotherapy. But please be aware, neither the specificities nor the levels of specific Ig antibodies can predict the clinical relevance of Ig sensitization, which means that the patient will really have a problem. This is very important because a positive Ig result obtained without clinical evidence will call for action. And the question is then, what will you do then? So if you have an accidental test result, you have to think what to do, because then all the action is on you, because it's like you are doing an MRI picture and you find a knot in the lung or somewhere else. And then you have to continue the differential diagnosis, but you also have to inform the patient about the test result and that it potentially can mean a life-threatening sensitization, but it can also mean that nothing will happen. And then you have a lot of things to obey. It's a legal issue. You have to provide the correct information, and you have to initiate also a cascade of follow-up tests to make sure if the sensitization is clinically relevant or not. So this is something that is important. This is also the reason why many doctors actually prefer to test for bee and wasp allergens only when there is evidence for clinical relevance, for example, after symptoms after bee and wasp stings. Now this is up to you how you will handle it in your practice, and it's difficult to provide here any, so to say, guidance, yeah, what you will do, but you should know about the problem itself. Now, having ended with a rather complicated lecture, yeah, I hope you appreciate that molecular allergology is progressing. There's a lot of it to be heard also at the next Immunology Congress to be held in a few weeks in Vienna. We have a masterclass on translational allergology that is dedicated to allergen molecules. You are invited to attend. Go to the Congress homepage, look at it. We have also, for interested doctors, day tickets. So it's possible to buy a day ticket and to pass by in Vienna and select, the program is already online, the day that you will eventually find very interesting. So if you are not interested in innate lymphoid cells, but you're interested in allergen molecules, you can select your allergy day if you want and attend for the day. Now let me end here, and I'm happy to take any questions you have, and I hope I didn't make it too complicated for you, but molecular allergy diagnosis, of course, advances day by day and year by year.
Yep. Let me thank you on behalf of all our participants for your lecture. As you said, it's molecular allergology. It's not easy, but it's the step forward all of us need to take now in better management of our patients. And coming to the beginning of your presentation, if you would let me have a few words, I think we can all see indeed the drug weight spreading and being more and more present in our population. And as Professor Valenta mentioned, it is a very important topic in our country, in Romania, and also in our region, because it's the transition of the patient's symptomatology starts to occur more and more in our practice. So I think what is even more important is to approach this allergen in the correct manner, taking care of the potential cross-reactivity and specifically in the patients that might have a polysensitization. And we need to thank you for sharing your knowledge about it and to make things more clear for us. And about clarity, I think it's one more time very clear for us that molecular diagnostic can and will make a difference in the quality of life of the patients, because I think this is the end purpose for all of us here. So I would put this especially in the context where there is immunotherapy available, it can be used, and by this way, we just make sure that the patient is suitable for the treatment and not wait maybe a few years and spend resources, financial resources on therapy that might not be very useful in the end. So one more time, I just got through this because it is some hot topic in our country that I can raise awareness of, and it's very important to take right actions. And now, one more time, thank you for your lecture. I found it very useful, and I'm sure that all our participants would agree with me. Now, if you're okay with that, we have few questions that we might answer to. So, let me start with the first one that came in the order. So, in case where a patient demonstrates positive diagnostic results for B venom sensitization, also has experienced several stings without any clinical reactions, is the initiation of the allergenic specific immunotherapy indicated, or are there any additional factors that should be taken in consideration before making this decision?
Thank you very much. I think this is an excellent question because it brings us to the real problem. And this would actually be when you have accidentally measured specific IgE against the bee and the wasp allergens, and the patient never had a symptom. Yeah, I think the next logic question is, when have you been stung the last time? That's very important. And then the patient can say, I have not been stung for many years, and I cannot remember what was before, yeah. Or I have been stung repeatedly, yeah, and I never had a problem. And in this case, I would rather advise to say, okay, we found this sensitization, documented, because you will have a discussion with your patient so that you have given the proper information. But if you have no problems, I would, in this case, not really recommend aggressive immunotherapy treatment. This is how I would handle the case. Yeah, some doctors may consider to provide the patient with an EpiPen. That's a possibility. Yeah, and of course to instruct also the patient of how to use it when it's the case. So this is a possibility. So perhaps the thing would be to say, okay, you didn't have, you had stings, you had no reactions, perhaps no immunotherapy, but I give you as a safety the EpiPen, and this is a way of how to use it.
Good. Thank you for your answer. I think it's all clear and it managed to touch the topic very fine. Next question. In a lot of works, it was shown that after successful immunotherapy, the specificity of IgG4 spectrum does not correspond to the specificity of IgE. Ig is produced to destroy pollen grain, but Ig4 is necessary to eliminate the allergen. Do you think it is necessary to use pure allergen for immunotherapy, or it is better to use broad mixture of allergens to produce polyspecific IgG4 for better elimination of native allergens?
Well, I think the question here, first of all, we must focus on what we have.
Yeah.
Unfortunately, we do not have molecular immunotherapy. Yeah? There's many reasons for this, and I do not want to dwell into it because it's like in the car industry. Yeah? As long as you can sell your old car, um, and there's no new technology coming, you will continue to sell the old technology. We have seen here that there is some allergen extract-based forms of immunotherapy that to some extent provide you the protection that you need, also to several allergen molecules. And we can now, with the molecular diagnosis, better prescribe. I myself think that molecular immunotherapy will be the future, and nobody will stop it. It will come. It will come in the form of hypoallergenic molecules that are also immunogenic and produce a lot of also polyspecific IgG4, but for allergens and not for other components of the extract. And this is also what is a little bit the core of the question. And this is we're injecting not only allergens, but also other pollen or other proteins with the immunotherapy. And then, of course, we get a lot of IgG against many different components, against things that we do not need actually. Yeah.
Yeah.
But at the moment, what we can say is we have to live with what we have, but we can learn to use it better.
Okay. Thank you for answering on this one. Let us go for the next. So, hello, Professor Valenta. Thank you for the webinar. I have patients with hay fever, and the results show that they are sensitized to ART-V3 more than to ART-V1. Will immunotherapy be successful in such a situation?
I love this question. It's excellent. Yeah. I think what we should do is we should do a similar study in rabbits like for the ragweed allergen extracts to find that out. Yeah. Because the only answer that you can get, there's two possibilities. We do the study and we find out the allergen extract-based vaccine that gives you also very good ARTV3 protective antibody responses. However, there's also another possibility that you, as the person you ask, can do. You can decide to select an immunotherapy product from a supplier um, with the products of which you were quite happy in the past, and then measure the specific IgG antibodies against ARTV1 and against ARTV3. And I personally would have a favor for subcutaneous immunotherapy. And typically, after the first course of immunizations, you should be able to see the increase of IgG antibodies against ARTV1 and against ARTV3. You can nicely measure this by, for example, immunocups with IgG conjugates. There is a ARTV3 cup, there is a ARTV1 cup, and then you can tell if the patient is producing the right antibodies. If not, you can change to another vaccine. So if this is a burning question and the patient has a lot of symptoms and you want to do immunotherapy, this would be the procedure that I would choose.
Perfect. Thank you for answering on this. We have two more coming. I will take the last one because it was related to the immunocap diagnostic. So, good evening. Thank you for the diagnostic of hay fever. What is better to use, individual molecules or Isaac?
It depends. It depends on reimbursement. It depends on your budget, and it depends also, I would say, a little bit on your skills. Yeah, when your skills are very good, yeah, you can save yourself quite some money. Yeah, by selecting in a clever way the single tests like the immunocaps. Yeah, uh, sometimes you have a very clear symptomatology, like seasonal pollinosis. Then you will know what is in the air. You have a handful of immunocap tests that you can run, and it will be done with that. Sometimes you have a very polysensitized patient, children with atopic dermatitis, food allergies, yeah, multiple sensitizations. Then the immunocap Isaac can save you some money, yeah, and it can bring you quicker to the decision. It also will give you all the overview at a glance. That's a nice thing. But in all the tests where there's many allergen molecules involved, the multi-allergen testing, I really say that a little bit less is sometimes more, because when you go to tests where you have an incredible number of different extracts or components on it, and then when you get a lot of positivity results, yeah, in the background level, yeah, where it's very hard to decide if it's something or not, yeah, it can bring you also into a difficult situation because then you do not know where you are. So it depends on the patient, and the deeper you are in molecular allergology, you can decide of what you want to choose and what helps you more.
Okay, thank you also for this answer. And the last question we have in the chat is, hello, I have patients, mother and 12 years old daughter with hay fever and LTP syndrome with anaphylaxis. Is it possible to use immunotherapy to improve the quality of life of patients with LTP syndrome? And again, thank you for the lecture.
I think this is also a very interesting question. Yeah, I've shown you these similarities between the LTP proteins. Yeah, and according to what we know and what we have seen is that the only little bit of similarity is between ATV3 and the lipid transfer protein from peach. Yeah, and I think there's very little cross-reactivity. So I would not think that immunotherapy, of course, because it also contains major pollen allergens. I do not know about the concentration of the LTPs in it, and I do not know about the immunogenicity for the LTPs. So I would rather have doubts that it works.
Okay, good. Thank you for answering to the questions. This was the last one that we had in the chat, and I want to thank to all our participants for asking the questions, being engaging, and participating actively to our webinar, because this is actually the way we can do it better to answer to the real questions we have from our daily practice. For the ones that would like to review or see any other materials, you receive a follow-up email to this presentation that you can rewatch. I would like to thank one more time on behalf of our participants and towards our participants for their presence here, and to Professor Rudolf Valenta for the very well structured and highly scientific lecture. It was not easy. It's not easy to digest, but it's real in the deep subject of molecular diagnostic, which makes a difference in our daily practice. So if I don't see any other questions, let me try to end this. Thank you again for your time, for your presence here. Thank you, Professor Valenta, for your lecture, and are willing to see you on our next session.
No, thank you very much for organizing this. I think it's very important. And it also shows that we are on a constant learning curve, and
Yep.
molecular allergology is getting better and better. Yeah. And we all can benefit from it. Yeah.
Absolutely, yes.
So
This is
all
the
a
progress
very
for them.
wonderful summertime. Yeah. So have a lot of good days. Yeah.
Okay.
And enjoy it. Yeah. And or continue to work. I prefer to work. It's the best time for me in the year. So all the best to all of you, and thank you for your attention. And bye bye.
Thank you too for your time. Have a good evening. Bye bye, everyone.
Bye bye.